Hello, students. Very good morning. So before we start this session, I would like to launch a technical poll. Please let me know if my voice is audible and the video is clear. Okay. Sorry. The poll should be on your screen by now. Please check it out. So let's start today's session. Okay. And today's class will have a very quick revision on very important topics because you have an exam coming up, so that it will help you out. Okay. So I'll end the poll. Yeah. So in the last class, we studied, I think, till transcription. And when it comes to translation, we have certain topics. And in these topics, we have, one second, we have certain important factors that we have to keep in mind. For example, when it comes to, yeah, the process of translation is very, very simple, like how you have studied in the textbook. But for the examination point of view, usually we come across the structure of tRNA, right? So that is very important, and the properties of genetic code. So we'll start with properties of genetic code, and then we'll move further. Okay. So when it comes to the general features of genetic code, so basically, in the beginning, well, while we talk about especially translation, okay, so we have to study about genetic codes. Probably you already know about genetic codes, but still I would like to insist on some of the points that you have to remember. So whenever the properties of genetic codes are asked, so we know that the codes are basically, triplet in nature. But if it traces the history of how the genetic codes were, discovered, like how they came into this, what do you tell? Uh, yeah, how they came into this conclusion that genetic code was a triplet nature. For now, this entire history of it is quite unnecessary. Hold on one second. Very good morning. Yeah. So let's get on with this. Today we are having a revision session, okay, so that we can finish some of the important topics with respect to two chapters. Okay. So yeah. So coming to the genetic codes, see the triplet nature was designed by specifically synthesizing RNA in an artificial system. So they understood that the codes have to be in 3 3 number. I mean, like 3 3 codes together. So then we have around 64 codes coding for 61 amino acids. And three of the codes are nonsense codes, such as here it is given UAA, UAG, and UGA. If you don't remember that, you can just remember it as UA, UAG, and UGA. Okay. So these codes do not code for any amino acid. And yesterday while discussing, I told you about methionine. AUG acts as a dual code. That is, it can code for methionine as well as it can act as a start codon. And I also told you about metftRNA, how this AUG acts as a special starter codon because the special type of methionine it brings near the starting of the translation, right? So other characteristics such as, genetic code is unambiguous. That is, the codes are very specific for a specific amino acid. That is only one amino acid. So the code is unidirectional. Like you have to always read the genetic code from 5' to 3' direction. And we know that the code is degenerate. That is more than you can see here. There is a pattern, proper pattern. For example, GUU, GUC, GUA, GUG. If you observe these codes, what do you see? The first two letters are alphabets are very, very similar, whereas the last ones are quite different, right? So even in alanine, it's the same. Even in glycine, it's the same. You see, there is a pattern that is appearing here. So what does it suggest? Even though these are different codes, they're all coding for the same amino acids. So that characteristic is called as degenerate. And next one, it basically do not have any punctuations while we read it. So these are the characteristic features of genetic code, and usually it is asked. Now, coming to tRNA. And the structure of tRNA is very, very important. See, when we study translation, whether in prokaryotes or eukaryotes, we target all the RNAs, that is rRNA, tRNA, and mRNA. All of them we have to understand to, to accomplish the process of translation, to understand the process of translation. Now, when it comes to tRNA, tRNA is very, very important because it is an adapter molecule and it is also having this 3D structure or three-dimensional configuration that helps it to behave it like that. Okay. So when we talk about the structure of tRNA is basically a three-dimensional structure. So just try to remember the labels of this diagram. So for example, we have a diagram like a clover leaf structure. So here, if a figure like this is given, how do you start labeling it? Remember that the short side of this arm is called as a 5' end and the long side of this arm is called as a 3' end. Now, the 3' end which is longer is called as amino acid acceptor end, or where the amino acids will come and join this. Okay. The left side of or the side towards the 5' end is the D arm. Okay. So on the 3' is a longer one, right? So you can see that it is T psi C arm. So how do we remember? 5 for D and 3 for T. Okay. So you have to go towards it. And then we have a variable loop. So basically these are the variable loop. And then we have one more loop called as anticodon arm. So each of it have its own significance. All right. So let's say what it is. When you see the 5' end, 5' end is very important. And why it is important? Because it has a special type of gynosin called as methylated gynosin. So here at the 5' end, there is a methylated gynosin. So how did it get a methane group? So when we see mRNA, we talk about something called as capping, tailing, and splicing. Remember? So we make the pre-mRNA to become the matured mRNA, correct? Similarly, even in rRNA and tRNA, what is happening is to make it more stable or to make it functional, what happens is a methane, CH4 group, that is methane group, is added to the 5' end of both rRNA and tRNA. So this process is called as methylation. So this, after the methylation, they become quite stable. So at the 5' end of the tRNA, there is methylated gynosin. Okay. Now, when we look at the 3' end, there are 3 nitrogen bases. Okay. Now here there is a nitrogen base, that is CCA, which acts as a code. Understood? So let's see here. So now, if you look at the loop, for example, this loop, anticodon loop. So at the other end, we have the codes. Basically, what they're helping us, these are the anticodons. And here is where it matches with your mRNA. For example, we know that AUG, is it writing? Okay, I don't have annotation. Hold on. Okay, you just understand here. We have AUG as a start codon. If AUG is present in our RNA, what will be the anticodon? Yesterday we solved this question. What will be the tRNA, right? A for U, and then U for A, and G for C. So these will be the anticodons that are present in the anticodon loop. So that is what recognizes and brings the specific amino acid here. Now, coming to the T psi C arm. So this is our T psi C arm, which is towards the right side. So they have a very unusual sequence. Basically, it is either thymine, or here it is there, pseudouridine and cysteine. This is very important. So the constituent or the contents of your T psi C arm are 3. What are those? One is a thymine, next one is a pseudouridine, and next one is a cytosine. So what is the role of this? So this T psi C arm, that is the arm that is present along the 3' 3' arm. So they basically help your tRNA to recognize the ribosome and bind to it during the protein synthesis. Okay. If you don't know the process, it's fine. We'll talk about it. So remember, T psi C arm helps in the recognition of ribosome. Okay. So that is very important. When it comes to DHU loop, so DHU loop has a specific constituent and we call it as dihydrouridine. So I know it is quite exhausting to remember all this, but this is necessary. Okay. I'll just summarize this. So this dihydrouridine, what do they help in? So the role of tRNA is to bring the specific amino acid to the location. Okay. So how do I say this? During translation, I think you already know the translation because your exams are already coming up. So during translation, we know that 1 tRNA comes at the site of translation and it gives the specific amino acid for the protein synthesis. It's just like a courier, right? So now, how do the tRNA recognizes that this is the amino acid that I have to be carrying according to the codes I have? So for that, DHU arm helps in sticking onto the specific amino acid. And also, how does it bring the amino acid? So this we have a separate process called as charging of tRNA, or it is also called as amino acylation of tRNA. So what does it do? This DHU arm, they have a specialized enzyme called as amino acyl tRNA synthetase. So it's given here, you see. This amino acyl tRNA synthetase, what it will do is, it will target the amino acid that it had to be bringing, and it will attach this amino acid to its structure. And that will happen through an enzymatic reaction called as amino acyl tRNA synthetase. Understood? So we have studied quite a lot about tRNA now. We'll just look into it. Now, as I said, our anticodon contains 7 unpaired nitrogenous bases. Among them, 3 acts as anticodons, which are complementary to the mRNA, right? So this is the structure of tRNA, the chlor-leaf structure of tRNA. So what did we study? You can understand this. What is 3 prime? Here is where the amino acid binds, and anticodon. Here is where the codes that are complementary to our mRNA. These two things are done. What is the role of D-arm? DHU or D-arm. So they basically help in recognition of our amino acid. What is the role of T-C arm? The role is to recognize the ribosome. What is the constraints of D-arm? It has a dihydrouridine. What is the role of T-C arm? The role of T-C arm is to recognize the ribosome. What is the constituent of our T-C arm? So here it is. T-C arm contains 3 important nucleotides. What are those? One is thymine and cytosine are common, but it has a pseudouridine additionally to it. What is there in the 5' arm? There is a methylated guanosine, right? There is already methylation happening to stabilize your tRNA. Done. So you understood the tRNA structure, right? So let's go further. So this is one thing important. Now coming to what is rRNA and what is its role. rRNA, several rRNAs are produced in your nucleolus region, and the role of rRNA is to make the constituent of your ribosome. So if you look at the ribosome, even though we approach ribosome as a cellular organelle, so here ribosomes are nothing but macromolecules, which are 80% made up of our rRNA and 20% are made up of proteins. So basically the rRNA's role is to make the constituent of a ribosome, so which helps in protein synthesis again. So coming to the translation process, you might know the translation process with initiation, elongation, and termination. And let's go directly into the process, okay? So the first process that happens with the, translation is by activation of amino acid. So how does the activation of amino acids happens? An amino acid attaches to our tRNA, okay, with the enzyme called as amino acyl tRNA synthetase that we just discussed. So tRNA amino acid complex is formed. Now, once this is done, now what will happen? You have, let me see if I have a diagram for it so that you understand. Yeah. Initiation. So 1 second, students. Okay, just give me a second. Yeah. Just give me a second. I'll just switch to the other screen so that we will understand this. Okay. Just 1 minute. We have 4. Yeah, it is here. It will be helping us with annotation so that we can study properly. So I'll just open a blank screen. Yeah. So that we can just write it down instead of just talking. So anyways, what I was talking about, I was talking about the process of translation, which is quite important. So yeah, we'll go textual wise. So basically what happens is you have, if you take, for example, eukaryotic mRNA, we have a 5 prime cap, correct? There is a 5 prime cap. And then there is this, structural gene that is mRNA. So I'll write it as mRNA. And then you have a tail, right? So now what is happening here? Now, once we have this mRNA, so the first thing what should happen is with our tRNA, our tRNA should go and bind to the amino acid. So how does this happen? So first of all, when we have a tRNA and the amino acid, the first step happens is amino acid goes and attaches to an ATP molecule. So when this is happening, here amino acid and AMP complex forms. So when amino acid and AMP complex forms, now it gives off 2 phosphate molecules. Here, by doing this, this will already carry an energy, a type of an energy that will help it to make an association with your tRNA. So that means if I break the bond between amino acid and the AMP, obviously it will release some energy, and we can use the same energy for making the bond between tRNA and the amino acid. Now, what will happen? This tRNA and the amino acid AMP complex will come together and it will become tRNA and amino acid complex. So this tRNA and amino acid union is powered by this enzyme called as, it is facilitated by this enzyme called as amino acyl tRNA synthetase. Okay, so you have to remember this because naturally we tend to forget some sensitive information. Okay, so this is done. Now next, what happens? Now once we have our tRNA, our target is to make protein, right? For that we have ribosomes. So our ribosomes are of 2 types. If you talk about prokaryotes have 50S and 30S ribosome, and eukaryotes have 60S and 40S ribosome, right? We already know this information. So these are smaller subunits. So the role of these smaller subunits is once you have a 30S ribosome, they go and attach. Okay, you have your rRNA. So this rRNA goes and attaches to the 30S ribosome. Now, your 30S ribosome comes and attaches to this cap region. So they basically recognizes this cap region, that is 7-methyl-gynosin cap in your eukaryotic mRNA. So once this is attached, now this will recruit your tRNA. So this tRNA, we know that it brings a special type of an amino acid, that is your initiator amino acid, AUG. So your first code will be AUG. So that is why it has to bring methionine, right? One second, it has to bring methionine. So the AUG gives us methionine. So now what happens, this special type of tRNA called as MET-F tRNA. So this MET-F tRNA means formylated methionine. So a special type of methionine, that is a formylated methionine, has to be bought. So the tRNA, I will write here, the tRNA MET-F is going to bring us formylated methionine. So once this comes here, now it is time for our larger subunit to come and occupy this place. Now we have our large subunit, we have our small subunit, right? And then we have our mRNA. So these 3 together will give us polysome. So remember this. So when they ask you what is polysome, don't get confused. It is large subunit plus small subunit plus the mRNA together, it is called as polysome. So once we have our phosphorylated methionine, and then we that is the tRNA, and then we have our small subunit, and then we have our large subunit together when they form the polysome. Now this complex is completed. Now from the cap region, they are dislodged to the AUG. That is the first base or the first genetic code that is present in your mRNA. From here, they'll jump to here. So this is what happens during the initiation. Now, coming to our elongation, right? Give me, yeah. Coming to our elongation. So actually there is some information, but right now it is not necessary because your exams are coming up and I do not want to confuse you with additional information. Okay. So once we have the elongation, now what happens? We have to understand the structure of our larger subunit. In our larger subunit, there are 3 sites and these sites are called as, so first one, you remember the A site, P site and E site. So what is A site? A site is called as amino acyl site, or it is also called as amino acid binding site. Usually it is amino acyl site. Okay. So now P site, it is called as peptide site. Okay. And E site is called as exit. So what happens here? Now, I told you already, a tRNA, which is carrying our AUG, right, that is methionine, is coming up. So when it comes up, it directly occupies the P site. Okay. So this tRNA, which is carrying methionine, directly occupies the P site. Now let us assume we have another nucleotide sequence that is coming up. Okay. So this nucleotide will have a specific amino acid, right, apart from methionine. Now another tRNA has to come bringing this particular amino acid to that location. Now when the amino acid brings this particular amino, when the tRNA brings a specific amino acid, it comes and occupies at the A site. Understood? So A site, what happens here, whether the right tRNA has come and occupied according to the specific sequence that is present in the mRNA is analyzed at the A site. Remember, still the tRNA is attached to the amino acid. Now, as this tRNA moves to the A site, now it gives off its amino acid. Now this amino acids becomes a part of the polypeptide chain. Okay, polypeptide chain. So once, okay, one second, polypeptide chain. So once it moves to the A site, now an empty tRNA comes to the A site and it is pushed out. Now this amino acid which is recognized at the A site moves to the P site. At the P site, what happens? Now the detachment of the tRNA with the amino acid happens. And also your larger subunit has an enzyme called as peptidyl transferase. So this peptidyl transferase will help us build a covalent bond, that is peptide bond between one amino acid and the other amino acid. So this gives the amino acid, right? So this has to be attached to the prior amino acid which was already there. So here it will cause us peptide linkage. So a peptide linkage is formed. So for all this, it's an energy deriving process. And what is required? You need energy. Okay, one second. Yeah. So for all this, what do you need? Okay. Yeah. For all this, what do you need? You need energy. For the whole process, gynosin triphosphate gives the energy, okay, for elongation. All right. Now, the same thing. A tRNA comes, recognizes its position, goes to the P side, gives the amino acid. Now, the enzymatic reaction takes place through peptidyl transferase with the help of GTP. Now, the peptide bond forms and a polypeptide chain keeps elongating, and then it goes to the E side. And this tRNA can be recycled. Okay, it can be again going to the different site where the protein synthesis is happening, and it can give the respective amino acid. So that is how it is happening. Did you understand? So this elongation keeps on happening in the direction of 5' to 3' all the way till the tip of your mRNA. Now what happens here? In the tail of the mRNA region, you have nonsense codes. So these nonsense codes are, as I told, UA, UAG, and UGA. So they do not code for any amino acids. So they're all called as stop codons. Okay. So these stop codons, since they do not bring amino acid, now let us imagine this translation bubble, which is having larger subunit, smaller subunit, and your mRNA. It comes all the way till here. Once it comes here, and if it recognizes any of these codes at the mRNA sequence, now what will happen is at the P site, okay, at the P site of your large subunit, okay, the stop codon is recognized. Once the stop codon is recognized, okay, a water molecule, H2O molecule, is added to the peptide chain, okay. At what end? At the carboxyl end. 1 second, people. I'll just get rid of this, okay. At the carboxyl end. What is carboxyl end? So here you have your, for example, N terminal, C terminal, R, and H. So this is the basic structure of your amino acid, just roughly. So this is the nitrogen terminal. This is the carboxyl terminal of your amino acid structure. So this carboxyl end, a water molecule is added by your peptidyl transferase, thereby terminating the process of translation and releasing the peptide chain, and then the entire polysome will disassociate, completing the translation. Ribosome can be utilized for the next translation process, and the mRNA that is being used for the translation is degenerated. Okay, so this is the process. Did you understand? So this is about the initiation, elongation, and termination of your translation. Now, coming up, we have one more topic. What is the one more topic? 1 second. Let me just go to the, yeah. So once we finish this, the what do you tell? Hold on. Translation, we have one more thing called as operon, lac operon. So when we talk about operon, so we talk about gene regulation, right? So when we talk about gene regulation, the gene regulation means either you start it or stop it, or increase it or decrease it. So this is called as gene regulation. That means a control unit of how this everything functions. Now, gene regulation happens at 4 levels that we know already. One is transcription, post-transcription, okay, and then we have transport, okay. So transcription level, post-transcription level, at the transport level, okay, and then we have translation. So what am I talking about? Transcription is where your mRNA, or I will write it as pre-mRNA, is produced, right? So here, either you can choose to start it or stop it. Next one, post-transcription is where the matured mRNA, that is by capping, splicing, and tailing, is produced. And transport is where the mRNA is moved from the nucleus to the cytoplasm through nuclear membrane, correct? And translation, we already know that mRNA is converted to proteins. So here, what happens is at all this stage, you can either start or stop the entire gene regulation can happen. Either you can choose to increase it or you can choose to decrease it. So these are the 4 checkpoints where all this is happening. Now, when we talk about your Jacob and Monod, who actually discovered the gene regulation in prokaryotes, so they told how can the gene get started or stopped, especially in prokaryotes. For that, they demonstrated with this model called as lac operon model. So what is lac operon model? So gene regulation we are studying in prokaryotes, that is called as lac operon. So in lac operon, basically we are studying the prokaryotic gene regulation in E. coli. Okay, sorry, E. coli. So what is happening here? So basically, if you have an E. coli cells, it uses glucose for energy, right? So now if the glucose is absent, it will try to look for alternate energy source. If there is alternate energy source, then it will be able to use that source. Now, to use anything, you need enzymes. And who will produce that enzymes? Genes will have that enzymes. So how do these genes get activated to produce a specific enzyme to break down the energy? I mean, to generate the energy is the whole point. So what is an operon? So if you talk about lac operon or a gene that is present in the E. coli, so here we have a promoter and then we have an operator, okay, operator, and then we have three sets of genes together, not one, two, three, lac Z, lac Y, lac A, and then this is the, I mean, actually, this is the terminator, okay? Anyways, now terminator is not necessary, so let's get rid of this. Okay, so this is the structure of the lac operon gene. Now, promoter, we already know it is a conserved sequences that helps in recognition of your transcriptional machinery, and it helps in the gene to function. Now, coming to the operator region is a specific sequence of the DNA which is sensitive to something called as repressor. So this repressor basically is a sequence of the DNA where it can attach to the operator region and block transcription. So this blocking of transcription happens whenever there is a repressor that is attached to the operator region. Understood? So how does it happen? So that depends on two situations. Understand? We'll discuss about it. So now this repressor region has a very high sensitivity towards operator. That is when it goes and binds to the operator region and block the transcription. Okay. Lac Z, lac Y, and lac A are three sets of genes that is coding for three different enzymes. For example, beta galactosidase, transacetylase, and then we have permease. Here, this transacetylase function is quite unknown, so we neglect this. Beta galactosidase and permease are two important things. That is, lac Z produces beta galactosidase, and we have lac Y that produces permease. Understood? So let us go back to the experiment. So first of all, in this whole experiment, for us, what happens is, one second, we have two situations. One, E. coli, when it is grown in glucose, and next one, E. coli, when it is having lactose and no glucose at all. Two situations, okay? Now, first we'll talk about E. coli when it has glucose. When already an energy source is present, it do not want any of the other genes producing unnecessarily waste enzymes. So, for example, we just discussed the structure of the E. coli's lac gene. So this lac represents lactose, and this lac gene is responsible for breaking lactose, which is a type of sugar, to give rise to glucose and galactose. Okay. Now, it already has glucose. When it has glucose, why does it need an additional energy source? So that is why it needs to block or stop the transcription that is happening in our lac gene. So how does it do it? Now, if you observe the lac gene, when there is glucose, obviously, this is your promoter, this is your operator, and then this is your gene that is having lac Z, lac Y, lac A. Okay, roughly I'm representing, okay, because of the lack of time. Now, this E. coli, when there is glucose, and what happens here? There is repressor. So this repressor should be free. When I tell free, usually what happens, if the gene is functional, this repressor is attached with allolactose. So whenever this repressor is attached with allolactose, the structure or the configuration of this repressor changes. When the configuration of this repressor changes, it can no longer recognize the operator region. When it can no longer recognize the operator region, it cannot block the transcription. That is why what happens, it will be free of allolactose. When it is free of allolactose, it will be having high affinity, or it can recognize this operator region and it can block the transcription. Once it blocks the transcription, now this gene becomes non-functional or dysfunctional. Understood? So this is when it is glucose is present. Now, if the E. coli is transferred from glucose containing medium to the lactose containing medium, where it do not have any glucose at all, but only lactose is present, what will happen? The cell will recognize that, okay, there is lactose, but no glucose. Now, what will happen? In the cell, there is allolactose. So this allolactose goes and binds to the repressor. First thing. So once it goes and binds to the repressor, now allolactose comes, binds to the repressor. Once it binds to the repressor, its structural configuration changes. Once the structural configuration changes, now it can no longer recognize the operator region. Now what will happen? Now the operator region becomes free. Once the operator region becomes free, now your transcription bubble can move forward. Once it moves forward, at the lag Z, it will produce beta-gal, and it will produce permease, and it will produce transacetylase. Okay. Once these three enzymes are produced, now what will happen? Okay. Now what will happen? This beta-gal will break down lactose into glucose plus galactose to produce energy. This permease, since our E. coli cell cannot directly let our lactose molecule to come into the cell, this permease goes to the plasma membrane, okay, or semi-permeable membrane. It binds to the receptors that are present in the plasma membrane and allows the lactose to come inside the cell. Once it comes inside the cell, now this engine keeps on running and your lactose will be metabolized. Again, in the glucose presence, the allolactose will release from the repressor region. It will go and block the operator region, thereby stopping the transcription. Did you understand? Yeah. So this is about lactose or lactopron. Now, coming to the next topic. Give me a second, students. All right. So coming to the next topic that we have to study very importantly is already it's HGP, that is Human Genome Project. So when it comes to Human Genome Project, the textuality and whatever is present, the facts, whatever is given in the textbook is of ultimate importance. So you have to really take care of every single fact that is given in your textbook. Very important. All right. So what is important in our HGP, the goals of HGP, and then the salient features of HGP is quite important. Please refer to that once, and I recommend you highly refer to that. Okay. So now coming to DNA fingerprinting, right? Now, what happens in DNA fingerprinting? Okay. So in DNA fingerprinting is a molecular technique to recognize two individuals from each other. Okay. Oh, sorry. Okay. So now what are we doing in DNA fingerprinting? So basically in DNA fingerprinting, we have a mechanism that helps us to recognize or differentiate from one person to another person. Alec Jeffrey in 1980s, he developed this technique based on polymorphic sequence that are present in our DNA. What is polymorphic DNA? So you might actually wonder what is polymorphic DNA. So the polymorphic DNA is basically the sequences that have VNTRs or STR, which is present in mini or micro satellite region. Okay. So in this mini or macro satellite region, mini or micro satellite region, so there are special sequences called as polymorphic DNA. What is polymorphic DNA? Polymorphic DNA is basically highly mutative sequences. So the number of repetition, like variable number, means the number of repetition of a specific sequence keeps on changing from one generation to another generation. So that is why we call these sequences as a polymorphic DNA. Now here, when we see this, how does it help us, differentiate from one person to another person? Now, when we look at the HGP, we studied that 99.9% of all human genome is same and the nucleotides is same. So that means all the functional DNA that we have, like genes, they are the same throughout the sequence. It do not differentiate. So it is impossible for us to identify who is who if we do a DNA analysis. So that is why R.F. Jeffrey thought going for polymorphic DNA is a very significant step in differentiating individuals. So how does it happen? So this VNTRs and STRs, as I mentioned, they are variable number of sequences. For example, I have my father and mother, correct? So I should be having a certain match. Yes, I do. But I do not exactly match either 100% with my father or 100% with my mother or even 50% or 100% with my sister. I'm quite unique. Like that of your physical prints that are present in your thumb or the fingers, right? Even in DNA, we have a very unique sequences. So these VNTRs, as I said, is repetitive sequences. For example, it can have 80, 80, 80, like around 250 repetition. Not only 80, it can be AG, it can be GC, it can be AC, anything, any of the nucleotide sequence which are repeating on and on for a certain number of times. And they are present, as I told you, micron, mini and microsatellite regions. So these sequences, if we find out in an individual, obviously we can catch the criminal. So what happens in the techniques or basically the principles? This is the principle of DNA fingerprinting. Now we'll go for the methodology. First of all, a crime has to be conducted. So let's say that we have three individuals, 1, 2, and 3. And unfortunately, the third person is killed. When the third person is killed, we have suspects. Let us say 1 and 2 are the suspect. And they have killed the number 3. So how do we catch number 3? So we go to the crime spot where the 3 is killed and we will do something called as swabbing. So when we do swabbing, we collect the samples of blood, semen, or any of the body fluids that are spilled at the crime location. It includes semen, it includes blood, it includes any of the body fluids that are somehow bruised. It can be swabbed in the nails, it can be swabbed in from the skin or the weapons that are used or the place they have fought or where the crime is being committed. Once we have the swabbing, next we have to do DNA isolation. So in DNA isolation, what happens? We do enzymatic digestion. So in enzymatic digestion, we know that the DNA is present within the cell, and when it is present within the cell, obviously we have to get rid of all the cellular debris that is present. For example, plasma membrane, the cytoplasmic constituents. So all that is digested through enzymatic. For example, we use proteases, carboxypeptidases, RNases. So all these ASs, which have the capacity to digest the certain biochemicals, are used to break down all these unnecessary macromolecules or micromolecules, leaving us only the DNA. The DNA is further isolated by spooling, whereby we add chilled ethanol and we isolate the DNA. Once we have the DNA that is isolated, we know that the DNA is very, 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 very long for the analysis. So we have to break down the DNA into small, small fragments. So this fragmentation, or it is also called as restriction enzymes, are added to your fragments. Thereby, you cut the DNA into small, small pieces. Now we have hundreds of pieces of the DNA. How do we analyze this? Now we label the DNA with any of the dye, fluorescent, or usually fluorescent dyes are used, so that it will help us visualize our DNA under 1 second. Okay. Now, once we have fragments, right, we have to separate, right? So to separate, we label our DNA fragments. Once we label the DNA fragments, we subject it to gel electrophoresis. Okay. So this gel electrophoresis, we use something called as agarose gel. So this agarose gel is used because it has to act like sieve, molecular sieve, gelati, what we call it as. So this molecular sieve has to help us, right? So the arrangement of agarose molecules are very uniform, like how your filter is there. So that is why we use agarose gel. So once we have an agarose bed that is being prepared, okay, we prepare a gel bed. So once we have the gel bed, we make small holes in the agarose gel bed, and we introduce our fragments into these wells. Okay. So when these fragments are introduced, for example, A's DNA, B's DNA, and the DNA that we have, okay, A's DNA and B's DNA are introduced. Now, what will happen? We introduce electricity towards it. So we know that our DNA is negatively charged and there is positive terminal on the opposite side. So we introduce DNA towards our negative side, and then we have the opposite as positive. Now, when we introduce electricity, high voltage of electricity, now the DNA fragment starts migrating towards the opposite pole based on its size. This is called a size expulsion, okay, separation. So once we separate the DNA fragments based on the size, we have the gel with fragments which can be visualized under autoradiography or UV light. We just put UV light and see if the fragments are separated. Now, these fragments have to be analyzed, like who is the criminal. So how do we analyze? Already from the crime site, we have something called as probe. That is the DNA that we have isolated from the crime site. So this probe is labeled, and this labeled probe actually goes and hybridizes. Hybridizes means it complements with the person. Like for example, if one person has committed the crime and we have a suspect, it will match with the suspect. Okay. So now what will happen? We have to make sure that this is for useful for analysis. So we take this DNA bar, or that is gel bar, and we go something called as Southern blotting. So in Southern blotting, okay, 1 second. Yeah. In Southern blotting, what we do is we make this gel bar, okay, we put this gel bar under nitrocellulose membrane. We take the gel bar, we take the nitrocellulose membrane and we place it one upon each other properly, and then we apply weight. So before we do that, we remove all the water from the gel so that the gel becomes like a paper. Okay, like the thick gel becomes like a paper. So once it becomes like a paper and we have a nitrocellulose membrane below it, now what will happen? The fragments will move upon to the nitrocellulose membrane, and this process is called as Southern blotting. Similarly, we have northern blotting for RNA and western blotting for proteins. Okay, so this is a technique. So once we have this, we hybridize this nitrocellulose membrane with our probe. Okay, this is called as hybridization. Now, once we hybridize, now what will happen from the crime side with the suspect? The DNA will start matching 100% with the fragments. Once we have a 100% match, now that proves that this person is a criminal who conducted the crime. So then we caught the criminal. Now we take the X-ray of the matched probes and we submit it to the court. So once we submit it to the court decides who is the criminal. Now the applications of DNA fingerprinting comes in several places. Okay. For example, analyzing samples of animals. Okay. In ecology, we use it. We use in forensics, that is investigating of crime, whether in murder or rape or any theft. Okay. And we also use DNA fingerprinting in solving maternal and paternal cases. So sometimes what happens, the DNA available becomes very, very less based on the crime. If the criminal is very, very smart, he might have wiped out all his traces or all the evidence that are linking to him. So what does he do? He basically erases it, like he's very smart. So do we have an option? Yes, we have an option. If the DNA sample is very, very small, then we go for PCR, which is called as polymerase chain reaction. Okay. So here we amplify the fragments of DNA, or if you have a very small amount of DNA, we make it more. So this reaction we will study in biotechnology. So this is not part of this chapter. So anyways, we utilize time in revising some vital topics that are important for your examination. So good luck Do well for your examination. Have a great exam season. Okay. I'll see you guys post examination. If you have any doubts, please post it in the chat box. You can discuss about your doubts regarding any chapter. And in case if you don't have, you can leave the session. Thank you very much. Have a nice day. Have a great weekend. Bye bye. I'll be in the session. Please put the message if you have any doubts.